Since we're talking about different experimental conditions, let's talk a little bit more about the Western blot protocol. It all starts with the protein extraction. The protein is going to be extracted from any different sample. For example, an animal tissue sample, cells that you grew in culture, bacteria, virus, virtually anything that has proteins, you can extract them from and run a Western blot. You will extract those proteins and quantify them. Then you will load them on a polyacrylamide gel, and in the presence of SDS and electric current, they're going to be separated by size. After that, you're going to want to transfer the proteins to a membrane. That step is very important because it would be very hard for your antibodies to recognize your antigen of interest in a gel. It's much easier to accomplish that on the surface of a membrane. Once you have transferred the proteins to the membrane, you will want to block the membrane. That step is important to prevent your primary antibody from non-specifically binding to the membrane. The primary antibody will then specifically target your antigen of interest. When you incubate your membrane with your primary antibody, that's what's going to determine the protein that you're going to be targeting, since your primary antibody was specifically designed for your antigen of interest. Then you will rinse that membrane to remove any antibody that is not bound to the membrane that would be giving you a non-specific signal. At that point, you're ready to incubate your membrane with a labeled secondary antibody. Now, the secondary antibodies can be labeled with all sorts of different molecules, and we're going to talk about that a little bit later. So you incubate your membrane with your secondary antibody that is labeled. You wash that membrane once again to remove any antibody that is not bound to the membrane, and then you're ready to image. The label that you have on that secondary antibody is what's going to determine how you're going to image that membrane. For more details on that, you can refer to our other Lambda U courses. For now, let's dig into our primary antibodies.